You are listening audio paper series with Yasir, and today's guest is Dr. Trampari from Quadram Institute, Norwich, UK, to discuss one of her publication, Exposure of Salmonella biofilms to antibiotic concentrations rapidly selects resistance with collateral trade-offs. It is published in Nature partner journals, Biofilms and Microbiomes. Welcome, Eleftheria, to Science Soundbites audio paper series.
Hi, Yasir. Thank you for having me.
Thank you for joining us. Would you like to introduce us to the project and why you studied Salmonella biofilm formation under antibiotic stress?
Yes, that's a good question. So, Salmonella is an important foodborne pathogen, which is responsible for several food-associated outbreaks, and that's why we used it as our model organism. Now, why studying antibiotic resistance in biofilms in particular? Because although we know biofilms are intrinsically resistant to drugs, biofilms' response to antibiotic treatment is poorly understood.
Okay. How you investigated the biofilm under antibiotic stress? This does not sound like a simple experiment to me.
So we developed the biofilm evolution model in the lab, in which you can grow biofilms on any material you choose and put them through a series of passages, watch them go through multiple life cycles, grow, build biomass, disseminate and colonize new surfaces. And you can do this under any evolution pressure you may want. So it gives you lots of opportunities.
All right. It means you have developed a biofilm assay model that you can use to study the effects of any kind of stress. So what are the main findings when Salmonella was exposed to antibiotic stress in your experiment?
So what we found, our major finding was that when biofilms get exposed to various antibiotics, they actually rise to the challenge and rapidly adapt. So they actually select for different mutations that make them more resistant to the selective pressure. And at the same time, though, however, we also observed that this resistance sometimes comes at a cost to other important phenotypes. For example, we show that biofilm formation sometimes is heated by this resistance to the antibiotics.
You mentioned various antibiotic stresses. Which antibiotics were studied in this project?
In this project, we exposed Salmonella biofilms to azithromycin, cefotaxime, and ciprofloxacin.
How Salmonella adapted to the stress of these antibiotics?
So first of all, under every selection pressure we used, Salmonella biofilms adapted, and they didn't only become more resistant to the drug they were exposed to, but they acquired, in certain cases, multiple resistances against different drugs. And this goes back to the mutations they acquired and how this translates to mechanisms of resistance that give resistance to multiple drugs at the same time.
All right. It means Salmonella develops cross resistance to different antibiotic classes when studied in biofilm evolution model. Can you give us an example from these mutations in Salmonella you have observed?
So one of our main findings, one of the mutations identified, they were actually multiple mutations identified within MZ. So MZ is part of a two-component system. It senses changes of the environment and it regulates the responses from the cell. So it's responsible not only for controlling membrane permeability and as a result allowing the drugs to be introduced to the cell, but it's also responsible for biofilm formation and hence the trade-off we observed. So they get better in blocking the antibiotics out of the the cell, but at the same time, they get worse in forming biofilms, which can cost them survival in the real world.
You mean when bugs become resistant to antibiotics, they become less efficient in making biofilm?
Yes, in this particular case.
This is very interesting, and thank you, Eleftheria, for sharing this amazing research paper.